All right, Unit 5, Part D, micro versus macro evolution. So when most people think about, evolution, they're thinking like this and how organisms have changed dramatically over time. Um, and yet when I was kind of hinting and talking a little bit about microevolution in the last presentation, and that's just a change within that population over time. And that's what we're going to kind of talk about moving forward. So micro means small changes. Micro is where the frequency of the trait changes within the population. I used that example in the last presentation on our peppered moths, right? Going from light to dark back to light, where over time, each generation, we had more light moths and less dark. And then when the environment changed, the dark were more successful. And then the environment changed again. And then the light were more successful again. That's microevolution. So mechanisms for microevolution, like how do populations change? How do they evolve? What does evolution mean? Okay, so that's what we're going to be talking about here. First of all, we have two scientists, Hardy and Weinberg, and what we call the Hardy-Weinberg equilibrium. So you have some people that are using, okay, exhibit A, B, C, D to support the theory of evolution. Well, these two, they said, well, basically, well, let's look at a different point of view. Let's say, well, what do we need in order to not have evolution occur? Okay, so in order for evolution to not, when we talk about to not occur, well, first of all, you need a large population. Two, we can't have gene flow. Gene flow means that we cannot have like one population gene, we're breeding one population with another population. We can't mix our breeding pools. So we cannot have the deer from McHenry mixing and breeding with the deer in Gray's Lake. Okay, that's gene flow, where populations are separated. Can't have mutations. No mistakes in our DNA replication or translation. And random mating. Completely random who mates with who. And no natural selection, meaning everybody is equally fit. Everybody is just as successful. We have the same adaptations, right? And if you think about this, you cannot have all five of those things meeting all the time. It's just not going to happen, all right? So this would be a hypothetical hypothetical situation here. It's not real. It's not going to happen. So if you cannot have all five of those conditions met, your population must be evolving. And they use math, and that's what they were using to support that evolution has to be a thing. Okay. So the process of adaptation, okay, this is where we have to adapt biotic and to biotic and abiotic factors to living things and sometimes non-living things. Could be weather change, could be climate change, it could be something that has to do with, the soil, okay? The amount of solar radiation, those are abiotic factors. The adaptation increases the fitness. Right? So when we talk about survival of the fittest, right, those that are the most fit for that environment will be more successful. So in order for populations, okay, for natural selection to work, when we're talking about populations, you have to have variation in genes. Everyone in the population cannot be the same. You have to be able to inherit those differences. The variations in success for the individual. So, for example, whatever is different, whether it is the color of your skin or whether it is the texture of your fur, those things have to give that individual some kind of selective or success in that environment. And variation reproduction. Of course, we're ideally in this situation, those individuals that have the best, that are most fit, have the best adaptation, will have more children. They will be able to provide by getting more resources, food, water, shelter, to then support their offspring. Now, there is this thing called genetic drift. All right, so we've kind of talked about natural selection, survival of the fittest, yada, yada, okay? Well, there is this term called genetic drift. Now, genetic drift is where you have, this is generally occurs only in small populations. This is where some freak accident, something happens, and it takes out a number of individuals. In this case, the poor brown frogs happened to be in the wrong place at the wrong time, and they got killed. They got taken out of the population. Now we just lost the brown gene, right? It had nothing to do with that brown gene being a positive or negative influence on that frog's ability to survive. It was just a freak thing that took them out. Founder effect. Founder effect's another situation where, again, we have a group of ladybugs here, and maybe there was some kind of storm, and all the red ladybugs happened to be on this tree log that floated out to an island. And now we're going to reestablish the populations of ladybugs with only the red ones. It's called the founder effect, right? Where a group of individuals starts a new population, but it's not, this population is not, right here, is not really representing the original population. There's no yellows here, okay? So this new population of ladybugs is going to be different in the color, in the variation of color in their population. Again, that's evolution. It's microevolution. An example in humans is that you guys know the Amish. Well, a very small group of people originally started this religious group. You know, they're very restricted. They don't allow strangers really into their population. And it just so happened that some of the original founding people had what's called polydactyl. Polydactyl is a dominant genetic disorder where you have extra fingers or toes. And so what happens is that particular population doesn't represent the rest of us because they are so secluded. Okay. So they have a higher number of individuals with polydactyl in their population than the rest of the world. Okay. Okay. So because they founded that population of humans and we're not, there's no gene flow, we're not mixing. Okay. So that is an example of a founder effect in human population. Another one is this retinitis pigmentosa. This is the same thing. We had a British colonizer. He carried a gene for this particular disorder. And so when he went to this island and had many relations and many children, there are 240 descendants on this island. And so they carry this gene for this particular type of it is a degenerative eye disease where they start to lose their vision. And so when we look at the population, the frequency that this gene is represented in this population of people on the island is much higher than, again, the rest of, you can say the rest of the world, right? So that is another example of this particular founder effect. And you can see here, so the frequency is 10 times higher than the original population that occupied that island. So before and after he got there, it's a lot higher now that, you know, once he started he brought his bad gene in to the population and then now it's fixed and it's much higher. So what happens is you usually start off with normal vision, and as the disorder progresses, your vision, your window of vision gets smaller and smaller and smaller and smaller, and finally you're completely blind. We also have what's called the bottleneck effect. And the bottleneck effect is where we have this whole population. You see all the different colors there, and we have some kind of mass catastrophe, whether it is a volcano that erupted or meteor. And only a few individuals, so you can see here, only a few individuals survive. And you can see where we've lost genes in this particular, from this catastrophe. So that's why it's called the bottleneck. And so it generally decreases genetic variation within populations. So here's genetic drift and the bottleneck. Again, genetic drift just means that there's some fluke or random event that decreases genetic variation, right? So we can see that that bottleneck effect here. We also have different types of natural selection. Right? So we have what's called directional selection, which is where an extreme phenotype is favored. And we saw that kind of like, right, where we favored the dark and then we favored the light. And so when we're moving from one side or another, like, we could move this way or we can move this way. It just means directional. We're going to one extreme, going to the black or going to the white. Okay, that's that allelic frequency shift. Right? We're moving in the direction of one particular phenotype. Some examples are bacteria and antibiotic resistance. So we know that we are driving antibiotic resistance with the overuse of antibiotics. And so with that, we are definitely selecting for, sorry, I'm plugging my pen back in. We are selecting for those individuals that have a mutation and the ability to survive with that antibiotic. Malaria, we're having the same problem now. We had this great medicine to help fight malaria. Well, it is a parasite. It is specifically a protozoan, and that protozoan we treat people with that medicine, and then those few individuals that have that mutation that allows them to survive, they are going to repopulate the next generations, and eventually now you're going to have a whole population that's resistant to malaria medications. Horses and size. Horses were small, and over time, directional and bigger, 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 bigger. Same with we got bears. Bears are also they're moving towards a bigger size. Okay. Stabilizing. Stabilizing means that we're in the middle, because not everything is black and white. All right. There are some, and we didn't get so much into there's some what we call non-Mendelian genetics, where like if you took a white and crossed it with a red snapdragon, which is a particular type of flower, you could get pink. Okay, we didn't get into incomplete and codominance so much. If you want to learn more about it, it is in your online textbook, and it is also there's Amoeba Sister on it as well. But this is where in some cases, like, we talk about skin color, we don't just have black and white skin, we have all kinds of shades. So in some traits, you have more than just two options. And for stabilizing means we're looking at the average of the trait. The average phenotype does the best. Okay. And disruptive is where we select against the average and the more extremes do better. So maybe white flowers do okay, and red flowers do okay, and pink, nope, they don't do too well. So that's disruptive. Now, we often talk about in disruptive snail color, and there are particular types of snails, and for this one snail color, like you have two different coloration patterns, and if you have a hybrid, it's kind of a mixture, and the mixture does not blend into either one of the habitats, so it gets easily seen and picked off by predators. Bird's eyes, you have large beaks that can crack the hard seeds, you got small beaks that can crack the soft seeds, but if you're in the middle, you have a hard time cracking those hard seeds, and you have a hard time manipulating the small seeds. So you don't do well with either type of seeds. Okay. So again, you would be better off being large or small in between. The struggle is real. Okay. So here's a picture of mice, and they're showing you the varying different types of fur color. So here's our original population. So if we were stabilizing, we would be looking for that average color in the middle. If we were directional, we were saying, oh, the darker one's doing best right now in that habitat. And if it's disruptive, we'll say, well, oh, the light and the dark do well, but in the middle, the hybrid does not. do well at all. All right, so those are the different types of natural selection that we see. And then we have maintenance of variation. Like, how do we maintain having genetic variation? Well, heterozygotes are really, really important for the maintenance of variation. They can carry recessive genes or sometimes because there are different types of inheritance patterns, they may show like a milder version of that particular trait. So for example, let's see what picture. Sickle cell anemia. Sickle cell anemia is a trait which you think, oh, having anemia is a bad thing. So you have your red blood cells. People with sickle cell anemia, they have normal shaped red blood cells, but when they exert themselves and the oxygen levels go low in their blood, the red blood cell sickles. And with that sickle shape, it can clog the capillaries and cause blood clots and other problems. It can't carry oxygen as efficiently, especially with all those clots. The body has to we can't fix it. We literally have to break down those red blood cells. So our red blood cell count drops, which is the anemia. Now, for this particular trait, African Americans have a relatively higher rate of this particular genetic disorder than Caucasians. And it was kind of baffling to scientists, like, well, why? Why is that? It's like, okay, we made this observation. Now, why is that? But what we found is in Africa, you have malaria. Okay, so if you are homozygous normal, meaning you don't have sickle cell anemia, the chances of malaria killing you are quite high. Okay, so those individuals tend to get picked out of the population by malaria. If you are homozygous, and I'm using two different letters here for sickle cell, normal and for normal, S for sickle. So if you're homozygous, you could say recessive. So you have two sickle cell genes, you will die from sickle cell anemia. It's a developing country. They don't have the medical care it needs to treat you. So you'll die from sickle cell anemia. Now, if you're a heterozygous, you have one normal gene, one sickle cell gene. You have mild sickle cell anemia, but you also get protection from malaria. So this group of individuals, yay, they are selected for in Africa. Okay, the heterozygous have what hybrid, I just forgot the word, but they are selected for. They have a definite advantage in that environment. We could call it hybrid stability. All right, so that's kind of a stabilizing effect, all right, in natural selection. So heterozygous individuals can actually be carrying genes that later can be very useful. Macroevolution, now these are adaptations that are just really big changes in an organism that allows for speciation. All right, when we talk about speciation, that means that maybe you had a population and now certain individuals are not going to breed with one another. Okay, they're so different, they're like, nope, not going to happen. And eventually, over thousands of years, they may not even be able to. So species, are organisms, when we talk about a species that can mate and produce viable offspring. That means if they have babies, they're living. They don't have stillborn, they're not dead. Okay. Now, they also have to be able to reproduce. Okay. So they have to produce viable and fertile offspring because it doesn't do any good if they have babies and they can't have children. Okay. So it's like if you cross a horse with a donkey, you get a mule. Okay. Now you say, well, then horses and donkeys, they must be the same species. They are not. Okay. Yes, they reproduce. Yes, their offspring is viable, living, but they are sterile. The only way you can get a mule is then by breeding another horse and a donkey. You cannot breed a mule and a mule. They are sterile. So you can't take a mule and get baby mules. Mm-mm. All right. So, you know, they are the donkey and the horse. They are two different species, but we are able to get a hybrid, but that hybrid is sterile. Okay. So that's what we're talking about when we talk about species. So population geneticists may look at it by saying if the group of organisms are going to share the same gene pool. So sometimes we may even say, okay, yes, I could theoretically take a deer from McHenry and a deer from Grayslake and cross them and get babies, and they're perfectly fine. But in reality, because of us, we have divided those populations up. Okay. The likelihood of a deer crossing all the busy roads and whatnot and making it successfully to Grayslake without getting hit by a car or something is slim to none. So, yes, we have different populations of deer. Yes, they are the same species, but they're not in the same population. So they're not breeding. So that separation from those populations over thousands of years may end up, they may be taking different directions in evolution, leading to speciation. All right. So this is where I'm going with the reproductive isolation. So this is where our populations are isolated. So maybe at one time, you know, in history, they were all able to migrate, had migration patterns and whatnot. But of course, thanks to us, they no longer do, and they are um, isolated populations of the same species that may, like I said, may eventually no longer be able to come back together and be one population. Some of these isolation, reproductive isolation includes structural. So maybe their male and female parts don't fit together anymore. Functional, all right, they may have different purposes. And behavioral. Maybe there is a certain ritual that a male or female has to do in order for during courtship to get that reproductive opportunity. All right. So we have a couple different types of reproductive isolation. First, I'm going to talk about prezygotic. Prezygotic means zygote is a fertilized egg. So we're talking about, okay, before we even get to a fertilized egg, before we even have a male and female coming together and gametes combining, okay, before that even happens, first we could have habitat isolation. Okay, so you could have organisms in different niches. Okay, we talked about those finches. Well, that one group of finches may have done really well with their beak shape to eat cacti. Others may have done real well for seeds, and maybe the plants that produce the seeds are on one part of the island and the cacti are on a different part of the island. That would give them habitat isolation. Temporal isolation is when they reproduce at different times of the year. There are some frogs where some frogs ovulate and produce eggs in March, and others it may be June. Okay, so if they're reproducing at different times, even though they are occupying the same space, they cannot get on the same time frame for fertilizing each other. Behavior, like I said, different courtship rituals. And then I talked about mechanical isolation a little bit. It could be the animal genitalia. Let me think about it. How in the world would a Chihuahua be able to mate with a Great Dane? Okay, it's not going to walk around with a portable ladder. Okay, so there are differences there that may prevent the sperm from being able to be deposited into the female. Flower structures, there's different types of flowers, and so that, you know, when we look at all the different shapes of flowers, that will also cause differences in plants. So plants have the same thing going on that animals do. And then we have what's also called gamete isolation. And that's where even though you have the egg and the sperm, they can't fuse. And you guys know in the spring, there's pollen flying all over the place from all the different trees. Well, I'm sorry, but if an acorn pollen lands on a pine tree, it's not going to fertilize it, right? So there are mechanisms in place so that the species can only, that pollen can only grow and fertilize its own species for plants, right? Post-zygotic is after. Okay. So this is going to prevent the hybrid offspring from being viable. So number one is zygote mortality. So chromosomes may be different. If you have a different number of chromosomes, once the egg and sperm meet, they can't line up their homologous chromosomes and they can't move to the next step, which is mitosis, right? You have a fertilized egg, it now has to undergo mitosis for 2 cells and then 4 cells and then 8 and 16, blah, blah, blah. We keep going on and on and on. If we don't have the right chromosome numbers, we can't move to the next step. Sometimes the hybrid is sterile. I talked about that with the mules. All right. So failure to correctly undergo meiosis. So even though they did, they were able to undergo mitosis and they were able to be alive. They can't, meiosis is not working for them for that to make either proper eggs or sperm. F2 fertilization. Sometimes the F1 is okay, but the F2 is not. Okay, we see that more so a lot of times in plant populations. Okay, again, if we can't keep the generations going, right, we can't maintain those changes within the population, and therefore we're not evolving. So modes of speciation. We have what's called allopatric speciation. We get the word patrick, if you think about someone that's a patriotic, like in the country, a country, a citizen. We're talking about that. Again, these are a lot of Latin roots. So when we talk about allopatric speciation, this is when we became actually separated. There is a physical barrier, okay, a geography difference that splits up the groups. And then due to mutation, genetic drift, directional selection, and all those other ways that populations can change by microevolution, over time, they become more and more different. And so first we have what's called post-zygotic, and then we have pre-zygotic reproduction isolation. Mhm. So these squirrels, this is an example of allopatric. So if you've ever been to the Grand Canyon, you know it's huge. And so we have these squirrels from the South Rim and the North Rim. There are 4 different species of squirrels. Of course, at one point in time, there was one, and then they live in completely different areas due to the changing landscape of the Grand Canyon over millions of years. And now we have 4 different types of squirrels. Sympatric is where a new species arises within a species without geographic isolation. And I always like to talk about wheat because everybody's kind of like, well, what in the world is going on with people that are gluten intolerant? And then you look at people like, are you choosing not to eat gluten? Is it a diet fad? Well, for many people, the wheat we are eating today is not the wheat that we evolved. And so with this change in wheat, many of us cannot properly digest it and it causes a lot of problems. So let's talk about wheat. Wild wheat can be either 2N, meaning you have 2 copies of every chromosome, so 2N is 28, or there's a different type of species of wheat that's 2N and has 14 chromosomes. The hybrid 2N is 21 is sterile. Okay, so this is where like because remember, if we go from 1N, right, in meiosis, right, we're going to reduce to 14, we're going to reduce to 7 to make eggs and sperm or gametes. Right, so if by chance those come together, right, 14 and 7 is 21. So we had 2 different species of wheat, their pollen, sperm come together, and we have a hybrid. Okay. Now the hybrid is successful in that it's viable, living, however, it's sterile. Now there is this thing that happens with plants sometimes, and they can have a mistake in meiosis where they don't have a reduction in number. And so if we don't reduce our number here with our 2N hybrid, we get 42 chromosomes. 42 chromosomes gives us a fertile offspring. Okay, so those hybrids can breed with other hybrids, but they cannot breed with a parental. We can't backbreed. All right, so I like pictures. So here is the species that we were talking about. So we have this species with 14 chromosomes here, 14 here. We have a hybrid, it's sterile. Okay. We have an error in mitosis. We don't have a reduction. So we doubled, we went to go through gamete formation, and then we didn't have our reduction in number. And so now notice how we have 2A's and 2B's. And this gives us our 28 chromosomes, right? 14 plus 14, 28. Now we have this other type of wheat here. Okay. So we take the wild one, right? And we're going to cross that one with this, and we get what we call a sterile hybrid, A, B, and D. And again, we have another error in our meiotic process. And now we have 42 chromosomes. Look at the size of that wheat. In plants, when we call this polyploidy. So polyploidy means you have more than 2 copies, right? So when we have polyploidy plants, many of them do really well. They're generally more what we call robust. Their leaves are bigger, their fruit is bigger. Notice here our seeds are bigger. This is our bread, right? This is our bread wheat. We're going to get a lot more wheat. Our yield will be much higher with this species than the other 3 that we talked about, right? Here, here, and here. Or even that one, 4. So the wheat that we're eating today is not the exact same wheat. It has evolved. Whether a lot of our polyploidy plants have had help with us humans, we have genetically modified them. So whether this has happened naturally in the field or been genetically modified, the wheat it's different. It's significantly different than the old wheat. And some of us are not able to process the proteins in that big, huge, bulky wheat. Cause a lot of problems with our digestive system, right? So macroevolution is where we have all these accumulations, these small changes that accumulate and become bigger and bigger and bigger over a period of time that eventually could lead to speciations. So again, species, this is when an individual again can breed and produce viable and fertile offspring. Right? So I'm going to talk more, when we're talking about macro, we're talking about going from fish to amphibians to reptiles. We've kind of been talking about that. So we got to understand the micro before we go macro. Right? So hopefully in this presentation, I talked about more types of evolution. I want you guys to focus on specifically the to the stop share. Specifically, when we talk about the difference between micro and macro and just some of the mechanisms on how these processes do occur. All right. And I hope I hope this is helpful. If you have any questions, please reach out and give me a holler. All right. I will see you in the next presentation. Bye.